CAS was formed in 2010, principally by Richard Burt Roberts, and it was built around sort of a couple of key themes, I suppose. It was built around the institutional expertise we had here in dating techniques, and particularly in luminescence dating, and the involvement of a whole bunch of researchers in some really exciting stuff going on in Southeast Asia, particularly on floras concerning the evolution of Homo floresiensis. And it sort of built out from there in terms along the lines of expanding the range of scientific applications we could use to answer questions pertaining particularly to issues in the evolution of humans and human behaviour. And the concept with CAS was to bring together researchers who worked across disciplines, but particularly in the sciences, working on these really sort of central issues about how human behaviour evolved and how we might use existing and novel techniques from the sciences to answer those sorts of questions. The great things about CAS is we've had this sort of rotating cast of really just exceptionally classy researchers working in different places around the world. So it's enabled us to become involved in a range of different parts of the world, a range of different sort of research questions.
So there's a long history in archaeology of using experiments to understand stone artifact variation in the archaeological record. The majority of this body of work was done by modern researchers replicating the forms of stone artifacts by using techniques and materials that we think were also used by past people to produce the artifacts we find. This approach, what we call a replicative flint knapping approach, is an extremely powerful one, and it allows us to address a wide range of questions relating to technical skills, learning, to economic decisions related to raw material, to also higher level questions about cognition and culture. But one of the fundamental challenges that we face in this replication process is one of confounding variables. So what that means is different variables or factors can potentially produce very similar outcomes in the stone artifacts, or the effect of one variable is conditioned by another variable that are also changing at the same time. So in flint knapping, the flake formation process is quite a complex one. We know that when we're replicating stone tools, a multitude of factors are involved at the same time. So the question therefore is how do we know the outcome that we're seeing actually relates to the parameters that we think is having an effect? To overcome this challenge of confounding variables, we need to be able to control the various parameters that are at work during the flint knapping process in our experiments. Because only by doing so can we be confident that the outcomes that we're seeing in the experimental process actually reflect the effect of the parameters that we want to test. We use a 3D milling machine to control the shape of the stone cores. In this case, this is glass, but we are also able to mill real stones such as chert and basalt. And what this allows us to do is we can produce multiple cores with identical shapes. In this case, this is based on the 3D model of an experimental Levallois core. We can then subject them to controlled flaking experiments using a mechanical flaking apparatus. We can then test the effect of individual flaking parameters such as the striking location, the platform angle, the striking angle, so for example the tilt of the core, to see how these individual flaking parameters actually produce variation among the stone flakes when everything else is held constant.
So I'm Malene Scotsmans. I am working at CAS, the Centre of Archaeological Science. I'm an archaeoanthropologist and I study funerary practices. So we are at AFTER, that's the Australian Facility for Taphonomic Experimental Research, and the public knows it as a body farm. So what do we have? It's UTS-led, and it depends on the UTS donor program. So people can donate their bodies after death to science. So there are two areas in this facility. One area is for surface depositions. That's where we study decomposition, we study taphonomy, we study decay, we study disarticulation, we study the bones. And then there's another part in this facility for burials, and that's a place where we bury donors and excavate them again after a set of time. But the reason why I'm here at the Body Farm is because I'm doing methodological research. We would like to understand different sequences of the funerary practices, and if we can read anything in the bones about those different sequences, the funerary sequences, the different steps before the body got buried. That's what I'm studying. What I also like is the lovely collaborations that you get at after. So with UTS, but also with different disciplines. We work with entomologists, we work with geophysicists, we work with chemists, forensic chemists. So it's that interdisciplinary aspect that I very much like about this site. What I also like, and I don't think it's done enough in my area of research, archaeoanthropology, is by studying decay and by studying bodies at the Body Farm or at the taphonomy facility, you kind of make a bridge between soft tissue and bones. So the archaeologists, anthropologists, they study bones, but the bridge between the soft tissue and the bone is sometimes missing. So we see that the decay is actually more flexible, or more, it's not as static as what we see in archaeology, and I think that's what I also like about putting the methodological aspect and the forensic aspect together with the archaeology and